So in this part of the assessment, I'll be discussing the process of which muscle contractions take place. So if we have a look at an overview here, firstly I'll be talking about the nervous transmission, which involves both the central nervous system and peripheral nervous system. Secondly, I'll then go on to speak about the neuromuscular junction, which is the gap between a neuron and a muscle fibre. And lastly, I'll be discussing the sliding filament theory. So if we put this into a sporting environment, the stimulus, for example, could be a ball coming towards you, and the sensory organ that would detect this would be the eyes, as they would see the ball coming towards you. And the impulse gets sent to the central nervous system via the sensory neuron. Now, the central nervous system is made up of the brain and the spinal cord, and its role is to receive the information and carry out a decision-making process and bringing about a response. This response is then sent to the effector via a motor neuron, which is in the form of an electrical signal. Specifically, the effector in this case would be the biceps brachii concentrically contracting to bend the elbows in order to catch the ball. So as you can see here, with the nervous transmission, it starts off with an impulse here in the form of an electrical signal, which is sent to the central nervous system here, which is made up of the brain and the spinal cord, as previously stated, and this is where the decision-making process will occur. The impulse then travels down an axon, which is this part here of a neuron, and it will specifically travel through these gaps here along the axon, which are known as the nodes of Ranvier. And this process here is known as saltatory conduction, which is the process of the impulse travelling through each node. And this will speed up the impulse, as you're not having to travel through the whole axon, you're just jumping through each node, which will get the impulse to the axon terminal at a quicker rate. So once we've reached the axon terminal here, we will reach the synapse, which is the gap here between the presynaptic membrane and the postsynaptic membrane. So as you can see, the impulse here will arrive at the neuromuscular junction between the end of a nerve and a muscle fibre. And in order for the muscle to contract, the impulse needs to go across the synapse here in order to stimulate the whole fibre. And to do this, the impulse will stimulate calcium ions, which will make the vesicles here, these blue circles, will make them go to the presynaptic membrane here at the end, and these are containing acetylcholine, which is a neurotransmitter. So the vesicles here release the acetylcholine across the presynaptic membrane and across the synapse, and will bind onto receptor sites, which are placed on the muscle fibre end. And if enough of the acetylcholine diffuses across the synapse into the motor end plate on the receptor sites, then the threshold will be reached and an impulse will spread along the whole fibre, causing a contraction to take place. And this is known as the all or none law. So if the threshold is reached, then the whole muscle fibre will contract, and if there isn't enough, then the fibre will not contract at all. So the sliding filament theory here is just a theory which explains how the muscles contract. So within each muscle fibre are different myofibrils, and within each myofibril is a sarcomere, lots of sarcomeres, and within each sarcomere are two myofilaments, one being actin, which is the thin filament, and myosin being the thick filament. So when at rest, the two filaments here, so the myosin and the actin filament, have no connection at all. The myosin, however, has attached to an ATP, which has been broken down into ADP and a phosphate by ATPase, and this breakdown of ATP releases some energy which the myosin takes up in order to be energized for when a muscle contraction needs to take place so that it can attach to the actin. However, the cross bridge can't be formed between the myosin and the actin because, as you can see here, there are these two proteins. One here is troponin and tropomyosin here, which block the actin binding sites in which the myosin needs to attach to. Therefore, there can be no attachment from these two. Okay, so when it comes to initiating a muscle contraction here, the impulse, as I've previously stated, will spread along the muscle fibre if enough acetylcholine reaches the receptor sites and stimulates the fibre, and it will travel along these T tubules here, as you can see either side. And this impulse here causes the release of the calcium ions from the sarcoplasmic reticulum, which is this light blue stuff here, as you can see in this picture. And this is where the calcium is stored. So the sarcoplasmic reticulum will release the calcium, and as you can see here, the calcium will then bond with the troponin, and this will change the shape of the troponin, which will cause both the troponin and the tropomyosin to pull away from the actin binding sites, therefore allowing the sites to be available for the myosin to bind to. Now, when executing a contraction, the myosin arms, as you can see in this picture here, the myosin arms here will attach to the actin and will use their energy from the breakdown of ATP to pull the actin towards the middle of the sarcomere. As you can see here, the muscle during contraction being shorter here, closer together than it is at the relaxation phase. And this is because the myosin heads have attached to the actin binding sites and have pulled the actin closer to the M line. Now this formation here is known as a cross bridge, and many cross bridges are formed between the myosin and actin in order to pull it along closer to the M line. Now this process will occur repeatedly so that the whole muscle can contract. Now the sarcomere length, which is the overall piece here, will shorten and get closer during the contraction, and the overall impact of this is the muscle fibre shortening, and the H zone here and the I band will get smaller, which is down here, the I band being there, and the H zone is the middle. The Z lines, which are these lines here, which the actin are attached to, these will move closer together because the actin is moving closer to the M line, therefore the Z line will move in too. However, the width of the A band remains constant as myosin does not move during contraction. It's only the actin that moves closer towards the M line. Now, when it comes to ending a contraction, the brain signals the contractions that are no longer required to the central nervous system, which will stop sending nerve impulses to the fibres. The calcium will then release off the troponin and they will return to the sarcoplasmic reticulum where they are stored for any further contractions that take place. The myosin here, in order to release from the actin binding sites here, the myosin will have to gain another ATP to release it from the actin binding site so that the tropomyosin and the troponin will return to cover the actin binding site, so no further contraction can take place. So now we are moving on to types of muscle contraction. So there are two types of isotonic contractions, one being concentric and one eccentric. We have an isometric contraction and lastly an isokinetic. So if we look at specifically a concentric contraction, this just means that when a muscle contraction takes place, the muscle length shortens, meaning the two ends of the muscles move closer together. So my example here is the upwards phase of a bicep curl. As you can see here, the biceps brachii are concentrically contracting, meaning the muscles being pulled closer together in order to produce the upwards movement. So when looking at an eccentric contraction, this just means that the muscle length will increase, so the two ends of the muscle are moving further apart. So in the upwards phase, these muscles were coming closer together and shortening, and in this movement, they're contracting, but they're also lengthening. So an example can be shown from the bicep curl, which in a downwards phase, as it is the biceps brachii that are still contracting, however they are lengthening as opposed to shortening. So an isometric contraction is where the muscle length remains constant, meaning that during contraction the muscle length will not change. So isometric exercises are done in static positions as there is no movement during the contraction. So for example, a plank here, the individual is still, so there is an isometric contraction of the muscles. And lastly, isokinetic traction is that when during contraction the muscle produces a movement at constant speed. So this doesn't usually occur in sporting action, so it's generally used to test and improve muscular strength, especially after injury. However, this can be shown in a breaststroke in swimming, so the water in this case will provide a constant and even resistance to the adduction movement of the arms. So from looking at all of these, it's clear that the eccentric contractions are generally to control certain movements. Concentric contractions are just to provide movements in general, and isometric contractions are shown to maintain movements. So this concludes the end of my presentation. Thank you very much.